So let's keep with some issues related to fish welfare. Now looking at the nutritional part. Some of the slides I prepared are overlapping a little bit with the previous ones, but I think it's okay. I will try to move fast in those ones. I will talk about some nutrients that can be used for improving fish health and welfare, but also about some previous studies that we did to try to improve fish welfare through nutritional strategies. I mean, welfare is a quite wide term. Frank this morning tried to comment a little bit on that. There are different ways of talking about fish welfare from the feelings-based scenario to a functional one, to a nature-based one, the 5 kingdoms. So I'm just focusing on the estrus part, okay, which is usually people think that fish are in a good welfare if they are stressed or not, which is not only that, but I will focus on that, okay? So, as you know, fish, when they are being produced, they are usually exposed to different stressors, such as crowding, high densities, handling, for instance, for vaccination purposes, or even transportation. Actually, there is not much knowledge about fish physiological responses after transport. There is some research that is going on now, and this will induce a stress response. And stress, as already Anita talked a little bit about that, can be acute or chronic. Acute is characterized by the fight or flight reaction, so the fish will cope with the stress and it will try to deal with it the best way possible, trying to recover homeostasis as soon as possible. In a nature-based scenario, the fish could go away from that scary moment, but fish are confined. Usually, they need to face with it, they need to cope with it physiologically. But then we have the chronic stress, so it meaning that the stress is more extensive, usually not that high intensive duration, but it lasts a lot. This will require quite a huge energy demand by the estrus process, so the fish will be coping with the estrus through the different physiological mechanisms, mainly secondary responses, and this will drop energy availability for immune mechanisms, meaning that at the end of the day, probably there will be some immunosuppression, which is classified as a tertiary estrus response. This is a classical diagram of how things happen in terms of estrus responses. They use the acute estrus response at the beginning with recognition of the estrus. There are some hormones that are reacting to that. We already heard about that. And then there is a transition with a secondary estrus responses. And then if the estrus persists, then we will have chronic estrus, and then we will see some tertiary estrus responses, such as, for instance, already it was mentioned here, the growth of the animal, behavior, reproduction, immune status, etc. So indeed, aquaculture intensification probably could have a negative impact in terms of farm profitability, translating to economic losses, and this would eventually need to adopt strategies to minimize these potential disease outbreaks, and sometimes it's needed to use antibiotics or other chemical treatments, which is not that easy because this needs to be strictly regulated, and only vets can allow that. Anyway, people knows about this. The consumer don't like that chemicals are used in the animals that are being produced, and this is neither environmentally friendly. So we need to find more alternatives to try to minimize as much as possible the use of chemicals in aquaculture. One alternative is the functional feed applications. As you probably know, you can use functional feeds for different purposes. You can use, for instance, astaxanthin. It's widely known as a pigment that will give that pink, orange, which is actually an antioxidant. So if you are eating salmon, it's good for your health, of course. We could also use functional feeds for improving growth, or we could use functional feeds for instance, for pigmentation in the skin. For instance, for carps, it's something that we done, and also for some aquaria fish species. We in the lab focus particularly in the enhancement of immune system, or immune status, disease resistance through functional diets. And this can be done through different supplements, such as pigments, nucleotides, prebiotics, probiotics, and amino acids. And I highlighted the amino acids because I'm going to talk about that afterwards. But it's important to highlight that it's immune stimulant and immune modulator. Immunostimulants are defined as a chemical drug stressor reduction that elevates the non-specific defense mechanism or the specific immune response. But it usually works, you see an effect after, but there is also immune to challenge the animal. The animal with a pattern. Have an effect on the performance of the animal. It's not allocated for growth. It's not recommended to do this for a long time. You can immunostimulate, for instance, on the strategy, depends on the compound you are using, prior, for instance, vaccination, prior handling for the grading process, transportation, whatever you think it's useful, but not for months. That would probably create a huge problem, not only in terms of growth, but we will exhaust the immune system, and that it would have bad consequences. I had a chat many years ago with a microbiologist, and they told us that also for humans, functional foods, we shouldn't use that in a daily purpose, let's say, because there was this yogurt that had probiotics that was very famous many years ago. Some of you didn't were still kids, but some years ago it was like it was called Actimel. I don't know if in Portugal you all know it. So you had one of those every day during weeks or months, and then you get the flu. Probably the flu would be much worse that if you were not using that or not eating that yogurt. Why? Because you were stimulating on the tiny immune system, so it was exhausted. And when the virus came, it was a perfect scenario for it. So be careful with immune stimulants. So there are many options in terms of immunostimulants that are being used in the market. There are many of them that are commercially available, and the industry is using them. For instance, levamisole, beta-glucans, alginates, even LPS in some cases. Some of them sometimes can be used as with other purposes, for instance, for vaccination. But we also can see amino acids, vitamins, and nucleotides as immunostimulants. In terms of beta-glucans and alginates, they are widespread and are found in plants, algae, bacteria, yeast, and mushrooms. There are some commercially available ones, for instance, Scretis and Igus. Igus doesn't exist anymore. It's not part of Cargill. Anyway, they use beta-glucans in some of their formulations. For instance, Biomari use sometimes bioactive alginates. Some know probably the most commercial beta-glucan is the Macrogar. It's something that is being used. It's from additive company, and then we can see sometimes the Ergosan as an alginate that it's used. Some Classical immune responses with beta-glucans, you can see in different fish species that usually increases the innate immune system. So you can see myeloid cells responding, such as increasing phagocytose, some lytic enzymes, also bactericidal activity, and sometimes it also increases resistance against pathogens. And in some cases, we also see antibodies production increase in the case of carp, which is interesting also for the acquired arm of the immune system. And with Ergosan, few studies are available, but you can see also similar outcomes with innate immune system increasing, and for instance, hits of proteins are increasing also in liver and gills in the case of CIIMAR, which is also interesting. But anyway, all studies are suggesting that immune responses and are varying depending on the level of inclusion in the diet, depending on the fish species, on the fish age, and the breed conditions. So basically, there are many factors that are affecting the effectiveness or not of these compounds, and that's why we have some collaboration with the industry. Anything we test at the lab, we should always prove it at the industrial level. Otherwise, we don't really know if it is working or not. Then we also have probiotics and prebiotics. I will not spend much time on this. You all know what is a probiotic, and you all know what is a prebiotic. So prebiotic basically is used by a probiotic to do the same effect. And usually in many studies in fish are showing that through different mechanisms, you can see an increased disease resistance by either using probiotics or prebiotics. In Europe, it's quite tricky, the use of probiotics because of the legislation. I think there's only one probiotic allowed to be used, which is the Lactocell, I think. But yeah, it's difficult and it's quite expensive. So unless you have something that is quite cheap on the one hand, and on the other hand, it's protecting a lot the animals, it's worth trying to do the studies to be approved by EFSA. Otherwise, it's not worthy. It's too much work and too much expensive. And there is also the concept of symbiotics, which is combining both, combining a good probiotic with a good prebiotic, and try to have synergistic effects. Usually, these synergistic effects can improve the survival and colonization of microbes in the supplemented diet, and also the activation of some metabolism from one or more bacteria with health promoting effects. But there can be risks, for instance, tolerance levels. We need to look at that. Maybe if we put too much, palatability constraints. This is something we need to think of when we are using an additive, that we are not doing any constraint in feeding behavior. And it's also important to have in account some issues regarding hierarchies, because depending on the species, sometimes there are huge, quite important hierarchies. For instance, in rainbow trout, you can see that there are dominant fish that eventually can ingest much more than subordinate fish, and this is something we need to take into account to avoid too much feeding of these compounds. I'm putting here just a practical example of the use of a symbiotic. This was done some years ago by the group of María Ángeles Esteban and the group of Miguel Ángel Morínigo in the University of Malaga and Murcia. And they use this symbiotic with inulin, which is a prebiotic, and this probiotic, which is Bacillus subtilis. They used in previous studies both compounds separately, and they were improving the immune status, immune response of CIIMAR. So we saw that, in fact, using this symbiotic, there was an increase not only in innate immune system, looking at the complement system, for instance, but also in acquired immune system. They saw an increase in IgMs in serum. And we also saw some T-cell markers and beta defensins, which is an antimicrobial peptide, that increased also in the animals fed with this symbiotic. So it looks like, okay, this is apparently a very nice potential additive to use for improving the health of the animals. So they induce a challenge, and they saw 100% mortality of the CIIMAR fed with this. So they were so wow. So there was a lot of things going on in terms of immune responses, but then the animals, all of them died. So they did an in-depth study, and we saw that the intestine was being affected. They saw a reduction, as you can see here, a reduction of the microvilli, significantly reduction, so a reduction in protection at the gut level, at the mucosal level. When they challenged the animals, the ones that were being fed with the symbiotic were more susceptible to the pathogen. So we cannot say always we see that, okay, there is some immune stimulation, there is some factors that are being increasing. It doesn't mean that you are giving protection to the animal. So this is something we need to be careful to think about always. Then I would like to talk a little bit about the amino acids. Amino acids, it's, as you know, they are the building blocks for proteins. So this is the basis for growth for proteins. They are also substrates for energy production. And they can be involved also in the biosynthesis of important molecules. For instance, there is increasing evidence that they are not only important for growth and energy metabolism, but also important for immunity, osmotic regulation, and reproduction. There is a lot of studies that are already reporting this, and there is a lot of knowledge about this. I would talk about a couple of them, for instance, which we have been working with them. Tryptophan, for instance, is the precursor of serotonin, and if you don't know, serotonin is important, it's a neurotransmitter. It's also precursor of melatonin, but in this case, I'm talking about serotonin because it's an important regulator of the immune response, the stress response, I mean, because it is the precursor of the adrenocorticotropic hormone that in the internal cells, it will be responsible for the release of cortisol. And the cortisol, as you know, it's a potent regulator of the immune response also. Sometimes a huge release of cortisol, a high release of cortisol will drop the immune response. So we have been studying the role of tryptophan in the bidirectional communication between immune system and neuroendocrine system, and how this, and also another thing important about tryptophan, it's in the deoxygenase, and the hydrogenin is a pathway which is quite important, at least when it is being used by immune cells in the response against in the downstream catabolism of tryptophan, because it will release some secondary compounds that are antioxidant and will also modulate the acquired immune system. So this is quite a complex study, because there are a lot of things going on, but we have been working with this, and I will show you a couple of easy studies to follow in terms of the use of tryptophan during stressful conditions. And then we also have been working with methionine, mainly PhD through the Senator methionine, because as you know, we don't know, but it's a donor of methyl groups and in different pathways in methylation, for instance, and it's methionine is precursor of cysteine, and it will participate also in the synthesis of glutathione, but also in the polyamine synthesis through the transformation pathway. And then synthesized, for instance, putrescine, spermidine, and they will be important in the proliferation of immune cells. So that's why we have been working with methionine. Regulatory effects when it's activated the methylation pathway, or some stimulating effects when we see the transsulfuration pathway being activated. But before I'm talking about those, I would like to tell you a story, because some years ago we have been working with flatfish and how different stressful conditions, for instance, handling or repeated handling, which will create a chronic stress, because we can see acute stress after a short event of stress, but if you repeat that repeated acute stress can become a chronic estrus. So we did that with the flatfish, and we also did the same with the high stock density, which is a persistent chronic estrus. And we observed that we measured the indispensable amino acids, the free indispensable amino acids in plasma, and we observed that actually chronically estrus by handling have a lower concentration in some of the amino acids, and we observed that the ones that have been stressed by chronic estrus, we saw the same drop in even more amino acids, and it was significantly different in the high stocking density ones compared to the low stocking density ones. So the theory was that probably these free amino acids were dropped, they were lower in concentration in plasma because they were being used. So if they were being used under estrus conditions, maybe we put it in the diet, we can reverse the effects or the negative effects. So we did the first study with repeated handling, again with flatfish. And we used, we have, of course, controls, a couple of diets for each group, control and handled fish. Handled fish was exposed 30 minutes a week during 4 weeks. We then two different diets, one high protein diet with increase in the history report, increasing digestible protein content translated into an increase in the digestible and indispensable amino acids. And we also added some tryptophan in there to try to improve the stress response of the animals. And we saw some things after 2 and 4 weeks. And we collected blood, brain, and it's a very small tiny piece in this kind of fish, and collected the liver for some proteomics studies. And we observed that actually, if you see here, we saw again the same drop in amino acids. In the green ones are the chronically estressed animals. I thought that it was reversed. So those amino acids in particular were recovered. We also looked at other parameters, and we observed, for instance, in the two-dimensional electrophoresis, we saw that there were some spots, in particular 172 spots that were reversed by the high protein diet at the liver level. In data probably is related to the degradation pathway of cortisol in mammals at least. This apparently also an increase in cortisol, as you can see here, the animals that were actually stressed chronically, not fed the high protein diet, they had an increase in cortisol level and also an increase in this enzyme, meaning that there was a higher rate of cortisol was not significantly different in the animals. As you can see here, it was not significantly different, but this is quite a heterogeneous response of the animals. So it was a level we see that is somewhat consistent. Then we needed to do it again. Then to see which ones are the ones that are playing a role here. So we work a little bit with arginine first, with flatfish. We also did it with sea bass, but we observed the opposite effect. So we dropped the idea. We're not doing it anymore. And with some wood, with animals in this case, the experiment was exactly the same as I said before. The animals that were chronically estressed, it was they were exposed to the air once a week or every day, depending on the experiment. But this one was once a week during 2 months, and we saw that estrus, which is the gray columns, had an increase in cortisol, as you can see here in this one. Not high levels, because cortisol levels usually are lower because there is a feedback, a negative feedback. But then we can, with the arginine, higher arginine levels, they drop cortisol. That 2 weeks, and this was after 2 months. And we see that after 2 months, we don't see that effect anymore. So we also looked at the blood at the end of the 2 months, and we observed that actually the red blood cells there was indeed no effect of the mechanism. That we see that how red blood cells increase in the stress animals. It makes sense because we were exposing them to the air. We were exposing the animals to some hypoxic events, so they increase the red blood cells as a type 2 of oxygen. And then we saw that in the blood, meaning that there was an immunosuppression of the animal, and the amino acid was. So in Senegal as well, we did a similar experiment, as I said before, but this time we exposed the animals every day during 2 weeks to air. It was 3 minutes air exposure daily, and they were .4, which is the requirement for this species, and then we increased a little bit the amount of arginine in the diet. 14 days of feeding, the animals were infected, and we also saw, we incubated some cells in vitro to see also some responses. But I would not exposed to estrus that were unstressful and fed the arginine diet. We saw that a decrease in the cumulative mortality. So the animals with a higher level of arginine survived more than the other ones. But interestingly, all the animals that were we thought, okay, studies in humans at least, that if you have a acute estrus every day, so be careful with that. That they say that he published an article very interesting that he says that a hassle a day may keep the doctor away, meaning that okay, sometimes a cold shower or organism sometimes it's positive. But I wouldn't do it anyway. It's interesting. It's a possibility that happened here, that maybe that acute stress, we are releasing catecholamines and other hormones that are also interacting with the immune system. Probably it allowed it to have a best way to study chronic stress. So we did another experiment, in this case with tryptophan, and we used high densities as a chronic stressor. This is a classic chronic stressor. We use densities that actually were .30 kilos per square meter compared to 12.5. The 31 kilos per square meter, the animals were like 4 layers of fish on top of each other. They were quite crowded. And then after 1 month, we look at some markers at plasma, and this is what happened. It was 40 days, not 1 month, and we saw that actually this was interesting, but it was also documented. We saw that the Low density animals had a higher cortisol level compared to the other ones, but this was because we were feeding the animals with tryptophan, and if you give to an animal cortisol, because tryptophan is the precursor of serotonin, and that is many years ago already saying that in rainbow trout this happens. Also, if you give tryptophan to an undisturbed animal, you would stress them eventually. And then we see a drop in cortisol in the ones that were at densities. We don't see that increase with tryptophan. Actually, we see a tendency to be lower in the ones with high tryptophan level. Alternative complement pathway activity in the animal, but it was reversed in the ones that were fed with tryptophan. And also, we don't see this clear here with the monocytes in blood in terms of differences between densities, but we see here an effect of the diet, which is interesting and points to tryptophan 2 as a more interesting treatment compared to tryptophan 4. And in fact, we also, when we see what happened at the disease challenge event, the animals were infected with phosphorylated cell pesticides, and we saw that this is two different scenarios that actually the highest level of tryptophan those are the high cumulative mortality, and in contrast with the control diet actually had what we see here is that there is a beneficial effect of tryptophan when they are not. This is like a double-edged sword. You need to be very carefully. And this is the land of Andorra, and we wanted to see what would be the role of methionine in the immune status inflammatory with two. We didn't work here with estrus and estrus fish because it was quite challenging for us in terms of mental design. So we just wanted to focus on the dietary effects, disease resistance. So this is the experimental design. We had a feeding trial for 2 weeks. Then animals were challenged with the photovoltaic cell interperitoneally. And then we had separate times where we could follow the time course response of the host against the pathogen, and also the cumulative mortality afterwards. And I will not go through all this data, which is a lot, so I will just comment about before infection. We observed that there was an increase in the neutrophils, in the circulating neutrophils, the neutrophils in the blood, in the animals fed with methionine 1, as you can see here, or not. You can see here that there was a higher level of neutrophils in the animals fed with methionine 1. There was also time effect, but it doesn't matter now. A drop in some genes that are related to T-cell markers. And so we hypothesized that there was like a dual role of methionine here. And also, since we observed an increase in neutrophils in blood, it was also playing a role through the transsulfuration pathway, being the kill donor to those polyamides I told before. So we see a proliferation of neutrophils. So this was interesting. So we wanted, and then we observed the response of the animal to a pathogen insult at the hematological level, blood, other things also. I will not talk about that, but for instance, at the blood and peritoneal level, meaning that we followed the number of cells in the blood and the number of cells that went to the infected focus. So we injected the pathogen in the peritoneal cavity, and we collected the pathogen, the blood that came, the cells that came to where the bacteria was. Then we also observed again that methionine supplement increased peripheral leukocytes, neutrophils in particular, and also at the peritoneal level, we observed also again total leukocytes being increased and also neutrophils being in the ones fed with methionine, which is to know whether the outcome in disease resistance was also satisfactory. And we observed that indeed the animals fed with both methionine diets showed a lower cumulative to the control ones, which is actually very interesting, and that's why we now in GRINNAQUA we are working with methionine in our package level. So then with an ectoparasite this happened yeah, maybe and this for today.